much indeed for that. And we're joined now by Tony, Colum and David, who are currently muted, but I hope that they will take questions. I know that we'll take questions, and I encourage you to put questions into the chat, and we'll try and raise them with them. Just maybe as a starting point, I suppose what I really got from all of you was the need to be flexible and responsive so quickly and respond to what was an emerging emergency. I mean, when Tony was talking about, you know, having to do manual tests, not being able to get the materials, and all of you talking about the resources and the supply chains and so on. I suppose, do you think that COVID has changed biomedical science? I suppose that's first of all a question for Tony, or is this something which is an event? Has it accelerated any changes in the way that we work together or we think about these topics?
Yeah, thanks very much, Duncan. Yeah, I think for sure. We had always envisaged the creation of a hybrid lab between the Western Health and Social Care Trust at Galvin and the University of Ulster that I had mentioned and Colum mentioned. It has vastly accelerated that, and as part of our Thrive project, part of the Derry City deal, the creation of that hybrid lab is now fundamental and integral to that project. The healthcare staff, what they do normally, and Colum alluded to the testing that is normally or had been normally undertaken. I think that the clinical colleagues now realise that within the university and academic sector, we're very adaptable and responsive to change, and it's what we do daily. And so there's a lot of benefit to be had from the multi-sectorial interactions that I talked about, and that is clearly evident. There's a kind of a perfect storm, I suppose, in the Northwest, if you like, in that we have a new medical school and we have city deals, and that comes at a time during the pandemic where we're in a position where we have the resources to implement some of the lessons we've learned from the COVID pandemic as part of our city deal. There is a risk that after the pandemic, people will fall back into their old ways. One of the things we saw during the pandemic was there was derogation of process, and that can bring its own problems where, you know, avoid the need for procurement. Obviously, we have to do things right, but what we have to do is do things fast. And that's something that, and I don't mean this in a derogatory way, the civil service and the health service are not used to doing things fast at scale, where people are fearful, you know, if I make a decision fast, I'll get into trouble, or during a pandemic, you must find rapid ways of responding that are consistent with good governance, and they're not mutually exclusive. And I think within the university sector, we do that extremely well. And the NHS have realised that working together in partnership with academia will be of significant benefit to not just to the NHS or to academia, but to society in general.
I'm putting the same question to the others. David, I mean, your own responses on how it has changed your own work?
Yeah, I mean, I would echo many of Tony's views. It's an unprecedented time to be a scientist and to see everything that's happened, in particular last year with development of vaccines and the many discoveries that are happening. So at an international level, certainly during my research career, there's never been a time where scientists have worked more closely together on a single event, on a single thing that's studied in science. So that was an amazing experience from my perspective. But I think the other important piece Tony's touched on and you were asking about is this idea of interaction between the health sector and academic scientists. And again, there's been unprecedented interaction between those two. And I think it's been really encouraging that both have been open to interact and to move forward on things quickly. And to sustain that effort is extremely important. we don't want to fall back necessarily. We want to have the agility going forward to act quickly, as Tony alludes to, for future pandemics, and even for the current one to sustain that effort is extremely important as we come up with some of these new variants of concern. So as part of that, there is a recovery group set up from the Public Health Authority, of which myself and Dr. Elaine Murray are involved, and other colleagues from other institutions, academic and healthcare sector. So that's an effort to try and sustain that momentum we developed during the pandemic between academia, other research labs, industry and the healthcare sector. And I think that's really important to continue, because we can see where the successful wins have been in developing some of the techniques, testing, vaccination and what have you. So it's really important we do continue that.
Colum?
Yeah, well, I think Tony and David have covered a lot of it there. I think it has been a unique experience, I think, for people like Tony and I have been, I guess, at the game a bit longer. I don't think we've ever had any period like this, but certainly it's of course true for many people. I do think it has, for me, certainly given me a great deal better insight into the clinical sort of the limits on clinical work and what sort of constraints people are normally working under, and I think a much better idea of what sort of things need to be done, and in some places the gulf, I guess, between the research and clinical approaches in many ways. So I think that's been a really valuable experience from my point of view, and I think from my clinical colleagues as well, who have seen perhaps that there are benefits as well to having a more flexible, open approach. But of course, I think here we had to be able to ensure that good governance as well is being followed, and as much as possible in the sort of crisis situation that was around. As Tony said, there was derogation of some things, but of course you have to ensure that you have got proper systems in place to make sure that samples are followed well and patient information is protected and so on. So for me, it's also been a huge learning curve there, but I think it's been a very exciting time as well.
Do you, I mean, this again is a question, interdisciplinarity was kind of evident in all of what you did, working collaboratively across sectors. Is there a tension in universities between pure research and you want to do pure research and this applied research, or is it now a case that those things are really very joined together and both have a role in the future of the way universities contribute to society? Give that one to Tony.
I mean, there is a tension which isn't real actually, because all of the basic research that Colum would do and David would do and our teams would do, they all have at the core translation to either clinical or commercial utility. Yeah. So the fundamental research that we all undertake, we always must continue to undertake basic research that is academically driven, and that is the bedrock that will feed the new clinical applications, the new treatments and the new tests. So if there is a tension, it's an artificial one. The two go hand in glove. If you don't do basic research, then you won't have any new applied tests, for example. Colum and we all mentioned the PCR test and the immunology test that David, those all started out, I remember Kerry Mullis was awarded the Nobel Prize in, I think it was 1988 or 1989 for the discovery of PCR. That started out as a basic research project. It was published in a relatively low impact factor journal. It has globally revolutionized how we do testing and molecular biology and genetics. So the two, it's three legs of the one stool. You have good basic research, good translation, and good application and impact. And the University of Ulster, our research office is called the Office of Research and Impact. So I think that exemplifies in summary, how the two go hand in glove.
Just a question and if you can answer it, I'll give it to David first, which is, it's the kind of what I would call the Donald Rumsfeld question, which is, what do you know now that you didn't know before this pandemic, and what now exercises you as a question that we really don't know enough about and we have to go forward? And of course, there are many things I'm sure you could answer that with, but what is, when you think now, a year on, what in your own research area, what do you think that has really come out that has been important, and what do you think we now need to move on to try to work through?
I think for me, with an immunology focus and thinking how the body reacts to the virus, again, an unprecedented opportunity to follow that as it happens in a massive population. So looking at the immune system's reaction to a virus as the population experiences that, and seeing what immunity is developed, has been really, again, something which is an unprecedented opportunity to myself as a scientist to capture that view. So, and the second part of your question, what would keep me awake at night? Well, I think obviously we're hoping that there will be a release from this pandemic, but we can see some of these variants of concern on the horizon, and it's just keeping an eye on those. And again, being aware, looking at some of the data emerging from England and Scotland in particular, we do see cases doubling, albeit there is some evidence of protection from vaccines, for example. So a big question remaining is just how stable that immune protection in the population will be, in particular to the current Delta variant and also possibly future emerging variants, and whether we can suppress it sufficiently, particularly in populations who've yet to be vaccinated for whatever reason. So I think it's the active monitoring of the immune response in individuals, in populations, has been, again, fascinating. And we need to keep an eye on this at a population level for new versions of this virus, if you like.
Colum, you want to answer the same question? What has been the kind of most striking thing that you think this pandemic has shown in your area of work? And then also what now we really do need to move on to find out?
Yeah, well, I think it's one of the things that did, I guess, surprised me at the start of this was that how little we could anticipate, or how little we knew about what the progress of the pandemic was going to be. And I think we still know surprisingly little about the self-limiting aspects of viral waves. I mean, I think we all are aware now of the different waves that we've gone through, and really, if you think about the first and even the second wave where there was no vaccination, those waves still come to an end on their own. They have a natural life course. And I think we know very little about what factors lead to that, and what is it about conditions that limit the spread or that cause the viral to go through its natural cycle like that, and what causes it to come back. I think those are really important questions that we know very little about. The other thing I think that surprised me a lot at the start was how little we knew about immunological reactions, and the fact that initially people were very concerned about vaccines not working because antibody responses seemed to be fading very quickly in the people that they were tested on. That has turned out not to be the case, thankfully. So I think that's a huge benefit. But I was amazed at how, despite very advanced healthcare in many of the countries that are affected, that we were still unable to really make solid predictions about that, and also that so many countries with very good genomics and really world-leading ability to sequence viral genomes, for instance, were still caught quite flat-footed in terms of testing, and we're not able to test at capacity at all, whereas other countries, say like Germany and South Korea, were able to test for viruses at a much faster pace, much more quickly. I think that was partly to do with the diagnostic industries that they had built up there. So those factors were a surprise to me at the time, I think, perhaps less so on the genetics front, but more and more general questions, I guess.
Tony, in terms of this personalized medicine you talked about at the very beginning, and genome sequencing and so on, has COVID-19 been an area where personalized medicine really has identified, I mean, I think the roles are clearly identified, but has really been important, or do you think it will be important as we go forward in terms of finding treatments, and in what ways?
I think it's at the heart of it. The term personalised medicine is sometimes used interchangeably with stratified medicine. And if you actually look at the way the government and the different countries have managed the COVID-19 pandemic, it has been a stratified approach in one hand, where you had people who were vulnerable, were stratified into groups that were told to shield. So that's stratified medicine in practice. We tend to regard personalised medicine as largely been focused on our genetics. And people say, genetics doesn't determine everything. The hand of cards that we're dealt definitely predisposes us to part of the COVID Genetics Consortium, an international study that we're going to submit our data to. It's already known that there are at least 5 genes or variants of genes that we carry that predispose some of us to severe COVID, and those genes are associated with, for example, genes associated with rheumatoid arthritis that David talked about. They're mostly associated with immune response. I said in my presentation that very soon we'll have our genomes available. You can get your genome done for less than the price of an iPhone. I think what this pandemic will do is fast track a better understanding in the public of what personalised medicine is. And it's not to say that current medical practice is negligent. Even with the application of current best practice, it's not good enough, because for 90% of the prescribed medications, they do not work in 50% of the people to which they prescribe. And that's not due to negligence, that's with the application of best practice. So, for example, if we carry a particular gene, for example, I mentioned one that might be associated with rheumatoid arthritis, we know that there exist drugs that are effective in treating arthritis, for example, that could have utility in treating only those people who carry that gene. They are the people who are likely to respond. If you think of the vaccine approach, the non-personalised approach is a blockbuster approach to drug development, and that's the general model that's practiced by the pharmaceutical industry, where they develop a blockbuster that's applicable to the entire population, and that's the business model largely. You can see already, even with vaccinations, you see discussions in the public domain about, vaccine A versus vaccine B having either more or less side effects. And you already have seen that the government have recommended that the younger age group are vaccinated with one manufacturer's vaccine over another, and that is based on the risk-benefit analysis at a stratified population level. Personalised medicine gets right down then to our actual genetics. And Colum is a world leader in his field in epigenetics, and it's not just the genes that we inherit. The troubles in Northern Ireland, for example, and Siobhan O'Neill will be talking about mental health. We know that if a woman is pregnant and she's subjected to stresses, severe stresses, or if people grow up during a famine, it can cause marks called methylation marks in our DNA that are transgenerationally inherited, and those are personal to each of us. So it's not just our genetic sequence, it's the marks that reflect our life experiences due to social pressures, famines, the troubles. And that's where I think there needs to be, while we can have the public health agencies and the research bodies can say, the whole world's looking at the genetics of COVID and so on, there will be a unique response to the people in Northern Ireland born of the experiences of, for example, the troubles that we came through, and that could have implications on how we how we respond to COVID and severity and so on, as well as our genetic sequence. So personalised medicine will be right in our places. People, individuals are taking control of their own health. They're getting their own genome sequenced separate from the health service. They're looking, and the problem is now you get your genome sequence today, Duncan, you go along to your GP and you say, there's my DNA sequence. He'll look after you as though you have two heads. The no profession currently do not have the infrastructure to deal with the train that's coming down the track of personal genomes, just in the same way as they weren't equipped to deal with the COVID pandemic, and they need to wake up and smell coffee.
Thank you very much. Colum, I was interested in your data and the presentation of the data in relation to the spread of COVID. I was wondering, is that the kind of thing that kind of data presentation something which has come from COVID, which actually we're going to see more of? Because it seemed to make a lot of sense to me as somebody who's not a biomedical scientist, but at least it gave me access to understanding how spread happened. I mean, has that been something which has evolved, the presentation of data in a way that actually is kind of accessible to the public, if you like?
Yeah, I think it is something that has sort of emerged. I tried in my talk to sort of give a little bit of background as to how I got into it. There is a lot of data comes in now through personalized sequencing, as Tony's referred to there. People get a lot of data back on their own DNA, and it was really from trying to handle large sets of data like that we got into this area. But people working in the healthcare system would already be familiar with the problem of dealing with large amounts of data from patient registers and from medicine registers and from pharmaceutical databases. So one of the major problems with all of these types of data is handling them in a secure way, but also in trying to make sense of them. There's so much of it, and you really, you're drowning in a sea of data, and you don't really know how to get what you're looking for out of it. And that was really the situation in many respects with the COVID pandemic. I think it came at a time where people have access to data. A lot of data can be generated very quickly on things like viral sequences, but making sense of the data in a timely fashion is really tricky. So what the brief taste there I give of the visualization method there, it comes from something that had been worked out partly by others prior to the pandemic. It was a freely available visualization tool called EpiContact, and it was used to try and visualize things like, say E. coli outbreaks, but it wasn't really designed to work at the type of scale that we were looking at here, where you got data on so many people at once. So really the team working with myself and with Damien at the PHA really had to come up with new ways to try and make it manageable, to try and plug in the data from Northern Ireland, from the contact tracing centre, and to pull in other data like test positivity and time since test and retesting and all of this. So really the trick is to try and figure out ways to interpret the data for it to make sense, not just to myself, but also to our clinical colleagues who are trying to do they have specific tasks they're trying to do, like they're trying to figure out, in this particular setting, is the spread really among workers in the setting, or is it coming in from outside? Is it people who are working close together who are having the main cause of spread, and are they bringing it home, and is it spreading when they're at home? So you have to be able to try and frame those questions in a way that you can then help them to visualize or to try to make sense of it. And I think that that whole area, sort of what we call big data or data analytics, while it already existed to some extent in the healthcare system, that has really been pushed very hard now. It really has grown a lot during the pandemic, and I see that not just here, but in England, Scotland, in America, and other places that are doing, in Ireland. that are doing very similar things, and that really has been a huge growth in that area, because there's a really pressing and practical need to be able to do that. Yeah.
Question to David, and then I think we're going to move on to the next section. But who knew? Did you expect that arthritis research and this pandemic would have a join? Was that something that, are you essentially understand yourself as scientists in immunology, or is this something that has kind of grown, this notion that research in one area can affect research in another?
Yeah, it's a really good question, Duncan. I suppose I would describe myself as a biochemist who's got an interest in an immunological disorder, particularly in arthritis, and most recently in rheumatoid or adult arthritis, but previously in juvenile. Could I have anticipated moving into the area of virology or looking at infection and immunity? Possibly. I started out in my early days at degree level doing a project on measles virus, and was kind of interested at that point. But specifically, I would say I'm interested in the kind of the protein side, the cell side of the immune system, rather than going from the virus side. But of course, the two have met, and as I tried to explain in the presentation there is some crossover, and it's very interesting when you look at some of these conditions, just the commonalities and the distinctions between what seems to be two very disparate conditions. You've got an infection from the outside, and the vector is the virus coming from the outside into the body, and the overreaction, if you like, as I described, of the immune system to that. Whereas arthritis is obviously quite distinct, and looking at a long, slow burn, if you like, of inflammation caused again primarily by genetics, but also the environment and the lifestyle of the person who has it. So it's been an interesting time for me as well to try and read up, stay abreast of virology, and again looking to the likes of the Delta variant, which we know has 12 different changes, but there's only 2 in that which are really key at the minute that we should be concerned about, and that goes back to Colum's talk again on the transmission. So transmissibility and the vaccine escape are 2 of the features which we really need to keep an eye on, and it's the vaccine escape portion of that which again aligns somewhat with the immune system. Being able to measure the body's reaction to new variants, I think, will be key in the future, and we can generate tests to try and do that simply by again taking chunks of the virus or these peptides and putting them against blood samples and testing whether individuals continue to have protection. So there's been a few studies around that, and it's something we would aim to do in the future with the research that we're doing at Ulster University.
Listen, this has been fascinating. I think actually we could probably do this for hours and hours and hours and do a seminar, and I imagine it's going to be true of all of them today. But I'd like to thank all 3 of you, Tony, Colum, David, both for your presentations today and the conversation, but also the work that you do. Ulster University is incredibly lucky to have the kind of integration we do have. I think it points to a really important crossover between what's done in universities and what is the concerns of society as a whole, and I think the work that you are doing will continue to be really important. So thank you very much for doing this today. Thank you for your presentations.